Isolation of Francisella tularensis outer membrane vesicles and their associated outer membrane proteins starts with osmotic lysis of bacterial cells. Logarithmic phase bacteria are sequentially treated with hypertonic sucrose, EDTA, and lysozyme solutions before being slowly diluted into deionized water. The resulting bacterial lysates are subjected to low-speed centrifugation, which removes cellular debris and unbroken cells, followed by high-speed centrifugation to pellet membrane vesicles. Next, total membrane vesicles are layered onto sucrose density gradients in order to separate inner membrane, or IM, and outer membrane, or OM, vesicles. OM vesicles can be separated from IM vesicles based on differences in buoyant densities, believed to be predicated largely on the presence of lipopolysaccharide, or LPS, in the OM. Immunoblotting of sequential fractions from sucrose density gradients shows spatial separation of inner membrane proteins from outer membrane proteins.
Hi, I'm Dr. Michael Norgard, and this is Dr. Jason Huntley from the Department of Microbiology at UT Southwestern Medical Center at Dallas. Today, Dr. Huntley will show you a procedure for extracting, enriching, and isolating Francisella tularensis outer membranes and their associated membrane proteins.
We use this procedure in our laboratory to study the outer membrane localized and potentially surface-exposed molecules for this intracellular bacterial pathogen. We have a busy day, so let's get started.
To begin this procedure, prepare and grow 2 500 milliliter cultures of F. tularensis bacteria as described in the accompanying written protocol. Retrieve the bacterial cultures from the shaking incubator and remove a 1 milliliter aliquot from each culture for OD measurement.